because we were in the Bayview campus, uh, adjacent to Johns Hopkins University, Bayview campus. We received a phone call from the ataxia telangiectasia clinician in Johns Hopkins, who said his one of his AT patients had a dramatic improvement on the behavior score because you need to have for periodic follow-up. And he was so surprised because that patient was always deteriorate, if we do not say stabilize. But that recent follow-up visit showed dramatic improvement on his or her working performance. Then the clinician in Johns Hopkins talked to the patients and the family says they were taking the nicotinamide riboside because it is commercially available as a diet supplement. So that really triggered Professor Ball and us to do a clinical trial in the USA.
Hello, Dr. Fang. You are a professor at the University of Oslo, where you run a lab studying molecular mechanisms of aging. So welcome to Modern Healthspan. Thank you so much for joining us today.
Thank you, Richard, for having me. I'm very pleased.
Thank you, Dr. Fang. So one of the things you study and is NAD plus, and in particular, how it gets impacted with age and what the kind of downstream effects of that are. So could you talk a little bit about NAD plus? Why is it so important, and what is the problem with it going down with age?
Yeah, NAD is from the it's initials from the name nicotinamide adenine dinucleotide. So NAD has an oxidized form, NAD plus, and reduced form, NADH. And this, we call them a couple, which originally considered as very important to regulate oxphors, including energy production through glycolysis, then went to the TCA cycle, and then to cut the fat beta oxidation as well as oxphors. So it's so important in energy metabolism, producing energy for me to talk, for you to host the meeting. And it has been proposed that we may die in some seconds to a few minutes if without NAD production, showing how fundamental this molecule is. And around 20 years ago, studies from Nanny Grante, David Sinclair, among the others, has linked NAD plus as a potential metabolite that is can slow down the aging process, because evidence was NAD is likely reduced when we're getting older. And when we pharmacologically or genetically reinstall NAD, we saw improved health span and increased lifespan in some model systems. Although regarding the way of lifespan extension, there are still debates.
So I did see there was a paper, I should have looked up the paper, that said that you can cut NAD levels significantly, maybe almost down to like 15%. And in animal models, in mice anyway, there seems to be no ill effects. Have you seen that, and what would your thoughts be on that?
I haven't seen that one, and I think I would look at that very carefully. It is reduced only in one type of cell or one organ, or it's ubiquitously reduction. And we should also look at what the parameters they did. So for I will use another way for example. So scientists in the field have been trying to using different genetic approaches to reinstall or slow down the consumption of NAD. And there are many NAD consuming proteins, such as PARPs. So in humans, we have around 17 PARPs. They are important to use NAD plus, to build the tree branch-like structure, enabling the DNA repair protein to anchor on this structure and facilitating DNA repair. And the second type are sirtuins, which uses NAD to deacylate target proteins like the master regulator of mitochondrial homeostasis, mitochondrial biogenesis, PGC1 alpha, FOX3, those things. And there are some other proteins like CD38, which is important in immune system. So among those three groups of NAD-consuming enzymes, some scientists have used PARP inhibition as a way to slow down NAD reduction in the aged mice. And they saw, NAD plus was increased and also increased metabolism, reduced fat accumulation. However, we are scientists traditionally trained on DNA repair. So we know how fundamental and important PARPs are. When you knock down PARPs, the animals may have a high sensitivity to like X-ray or sunlight or UV-induced cancer. Even you may just knock down PARP1, which contribute to 90 to 95% of the function. You have PARP2 and the others to complement partially. But under extreme conditions, I think those animals may have a potential chance to get side effects. So this is, if I go back to this NAD reduction to 50% on how they impact the quality of life in the animals, I'm not sure whether they checked all those parameters or they just checked a couple of few. Okay, the running speed is okay, but how about extreme conditions? If you let the animals run for 2 hours, should those with less NAD runs the endurance and resilience as good as the wild type? We need to take a look.
Yeah, while we were talking about the enzymes that consume NAD, right? The PARPs,
Mm.
the sirtuins, and the CD38. So it seems that reducing CD38 would be perhaps the optimal approach rather than, like the NAD repair enzymes. You kind of need them, I think, especially for long-lived animals like humans. Would you agree with that? Would CD38 be a better target?
Yeah, so I had my postdoc training 2012 to 17 with Professor Wilhelm Bohr, a Danish American professor. We first worked with Chromedex to do the NAD-based experiment on the premature aging diseases. And then we were approached by, I think, a big pharmaceutical company to work on CD38 inhibitors. So we had some preliminary results. Because in C. elegans, it just seems C. elegans is a major model we have been using. It just seems there is no CD38 off-log in C. elegans. So the data were majorly in cells, and a lot of behavior experiments we were unable to do. Uh, I understand Professor Eric Verdin, his team, and others have done a lot of work on manipulating CD38 expression or inhibition as an approach to save NAD and check whether it was able to improve the endpoints they are interested in. The data look very convincing, and I were in different meetings with Professor Eric Verdin, so we also talked about that. Uh, there was a very recent study by uh, Israel group showing the importance. I think it's Michelle. She is an immunologist, so she shows how to inhibit CD8 as a way to increase memory in Alzheimer's disease, like mouse models. So those data are very convincing. But there was one thing we needed to keep in mind. That was in early 2000, there was a team in Japan which showed when the mice with knockdown of CD38, they had reduced the capacity to interact with each other. The social activities were low. So I just wonder, scientists who are working in this uh, should also check whether knockdown CD38 in their case can impact the social activities of the animals.
Yes. Well, yeah. So that's like a knockdown. So a knockdown would remove it completely, right? Whereas if you
Yeah,
pharmacologically, you'll, I guess, reduce it without
yeah.
hopefully removing it completely.
So pharmacology could be more safer if the target protein also has some physiological activities in cells.
Yeah. So, sorry. So back to NAD and aging. So
Mm.
have you done kind of preclinical trials on NAD with and aging? And which precursor did you were sticking with NR, or have you used other precursors?
Mm. We worked with Chromadex through MTA, between Chromadex and the National Institute on Aging. So we worked on NR. I think before 2018 or 19, NMN was very expensive. So that was why we got a lot NMN from Professor David Sinclair. We collaborated on a cell metabolite paper in 2016, and I think it was very expensive. So we used both for our experiments. I want to share with you, we're very proud that it was around 13 years work from understanding of mechanisms to preclinical evidence in C. elegans mice and these premature aging cells, then to clinical trial. Uh, because it may be time consuming and also very long for you to do a biological aging, not only in like humans, but even in mice, it may take very long. So then we have a niche to work on a group of disorders called premature aging or accelerated aging disorders, such as Werner syndrome, due to mutation of a gene Werner, which encoding a protein Werner, participants in DNA double-strand break in DNA double-strand break repair. Ataxia telangiectasia, which is caused by mutation of a gene ATM, ataxia telangiectasia mutated, encoding a protein with the same name, which is a master uh, player in DNA double-strand break repair, including both NHEJ and HR, as well as xeroderma pigmentosum group A. So except Werner syndrome, all those other premature aging disorders show very severe neurodegeneration phenotype. And Professor Ball, my former mentor, uh, we were interested in addition to unrepaired DNA damage, are there any direct or indirect or secondary drivers or risk factors that can exacerbate the so severe neurodegeneration? And with this in mind, we performed metabolomics, proteomics, and different approaches. Then we narrowed it down to NAD plus because our data show it was dramatically reduced in the cell lines and animal models we used. And however, if NAD is reduced, it could be a bystander, may not necessarily a culprit of the disease. So to test this hypothesis that it could be a risk or potential by a culprit, we just pharmacologically giving NAD precursors, including NR and NMN. Sometimes we also compared nicotinamide as well as NAD plus itself to animals. And we were able to see dramatic extension of lifespan in the AT ataxia telangiectasia mouse model. In the vehicle group, they normally die around 3 to 5 months, but with NR treatment started from weaning status um, continuously until their death. we saw the lifespan extended to 8 to 10 months. And at the same time, we also had the wild type control. So for the wild type control with this 1 year more, I think we followed up. Um, we also saw around 5 to 10% extension of lifespan. So it's not so dramatic in the wild type, but we saw an extension. And the piece of data of the wild type mice was in line with the science paper published by Professor Johan Oinks from EPFL. So in that study, he showed in middle-aged mice or old-aged mice, 20, I think around 2 years old, treated with NR, he saw improvement of lifespan, like 10%. But the focus of that paper was on improvement of stem cell stemness. So, and after I moved to Norway almost 8 years ago, with my head of department, former head of department, Professor Hilde Nielsen, who was also in the original clinical paper in Cell and Cell Metabolism. We really wanted to test whether it works in premature aging disease, AT. In Norway, a country with a small population of 5 million people, we have around 27 kids registered with this disease. And we were able to recruit around 13, and there were 10 kids, some may not kids, may adolescents, so less than 20 years old, joined the clinical trial, but 10 were able to follow up for 2 years intervention. Among the 10 participants, 9 showed a dramatic improvement in their clinical performances. And this paper was published in 2023 in like Behavior Movement journal. We were proud to say that uh, actually before we published our results, there was a team in the Netherlands. They saw our preclinical data. It's a big team on ataxia-telangiectasia AT. They had, I think, 3 months or 5 months treatment to around over 20 participants, and they saw dramatic improvement of behavior. A recent study is one case report by a hospital in Austria. They showed that a 3.5-year-old boy, when he went to the clinic to see the doctor, he was unable to draw the line in a cartoon between the hand to the balloon. So the task you need to draw the line between each of the fingers to the balloon, and his performance was terrible. And only after 9 months treatment, and this boy was able to draw a very beautiful straight line between one hand to a balloon, one hand to a balloon, because it was in his development stage. So definitely the authors also discussed that we may not exclude, because his development stage can also benefit on performance. But if you put all those 3 clinical studies together in 3 different European countries, we were actually very positive. So that was from our preclinical to clinical. And there was a very interesting story on why we really want to do the clinical trial. It was, I think, after we published our cell metabolism in 2016, maybe after half a year or few months, one day Professor Wilhelm Bohr at the National Institute on Aging at NIA, because we were in the Bayview campus, adjacent to Johns Hopkins University, Bayview campus. We received a phone call from the ataxia-telangiectasia clinician in Johns Hopkins, who said one of his AT patients had a dramatic improvement on the behavior score, because you need to have for periodic follow-up. And he was so surprised because that patient was always deteriorate, if we don't say stabilize. But that recent follow-up visit showed dramatic improvement on his or her working performance. Then the clinician in Johns Hopkins talked to the patients and the family says they were taking the nicotinamide riboside because it is commercially available as a diet supplement. So that really triggered Professor Ball and us to do a clinical trial in the USA. But we were unsuccessful in getting funding to do so. So then ended up we started to do that in Norway.
So a couple of questions in there. What what dose was the NR, and
Mm,
what do you think is the mechanism? Because we talked like NAD impacts a number of different things.
yeah.
What do you think the NA? What do you think made the difference for these people?
Mm. So the dose we used for the kids and some are or adolescents started from 100. My number may be a little wrong, but it's in that level. We had a gradual increase. It's like 125 milligram per day. Then we did this for 1 month, then increased to 250. So and then finally went to 500. So then it's a gradual increase. And we followed up all the parameters. A few reported side effects like the counter, uh, I think how to say the watering poop, the
Diarrhea.
poop, yeah, diarrhea, and a few may have the itching and flushing. So that's common side effects of nicotinamide. But those are reasonable and they were able to handle it. Especially those diseases are very predisposed to cancer. However, after 2 years, treatment, we didn't see any sign of cancer. That could be we can have another topic at the end. And you are talking about the molecular mechanisms. So in the preclinical experiments, we so the long story in short was it was able to increase the homeostasis of mitochondria, increase the quality of mitochondria through the induction of mitochondrial autophagy, termed mitophagy. So NAD plus stimulates mitophagy to recognize and engulf and recycle the damaged mitochondria. This is the number one mechanism we proposed and evidenced. Number two, for those premature aging disorders, they have compromised DNA repair pathways. For example, when we work on neurodegeneration, um, neurons are post-mitotic cells, means they do not replicate. Then for the DNA double-strand break damage, uh, they only can use NHEJ, not HR. So HR is used for replicating cells. And we checked NHEJ in our systems, and we find with the ND plus Prixer treatment, we're able to see increased NHEJ performance with the mechanism that 31 uses NAD plus to deacylate Cu70. Cu70 is important protein participants in HEJ. Deacylated Cu70 is essential for its recruitment to the double-strand break site of the post-mitotic neurons, and finally to increase the DNA repair efficiency. And we did this in both, I think, neurons as well as in the C. elegans models.
Right. And NHEJ is a double strand break fixing enzyme.
NHEJ is an initial of the term non-homologous end journey.
All right.
It's one of the two types of pathways to repair the double-strand break damage.
Okay, so since we're talking about mitochondria and the importance of mitochondria, so one of the things you look at, I think, in your lab is dementia, particularly Alzheimer's disease,
Yeah.
and the importance of mitochondria and mitophagy in
Mm,
that. So, why is mitochondria in, I guess, in neurons so
mm,
important for kind of the pathway to dementia?
yes. Uh, so the the weight of our human brain constitutes 2 to 3% of our body weight. However, it can consume up to 30% energy. It means our brain needs a lot of mitochondria and also in high quality to produce 10 times of the ATP uh, per second on average compared to other organs. This statement may be wrong, but it's just to show we need a lot of ATP for the neurons to do the activities they are doing in every seconds. And uh, the focus on mitochondria are based on the below uh, evidences. So in addition to be interested in those premature aging disorders, we are also working on the common age predisposed disease. Um, because our way is premature aging, they are important, but still an orphan disease. Not so many people have the disorder. So we wanted to use the discovery from premature aging and see whether this mechanism, like reduced NAD31 mitophagy excess, can also be applied to common aging predisposed neurodegeneration, using Alzheimer's disease as an example. So to test this, we collected samples from Harvard Brain Bank. We had a careful and serious evaluation on the pathologies using electron microscopy. So in addition to see accumulated A beta plaques and tau tangles, those are disease-defining pathological features of Alzheimer's disease, or we call the brain garbage. We also saw accumulation of damaged mitochondria compared to sex and age matched controls. This accumulation of damaged mitochondria in the hippocampus and entorhinal cortex, those brain regions important for memory, are reported before. We were not the first one. And scientists in the field have proposed increased oxidative stress as a cause of mitochondrial damage. However, there are many studies if you use antioxidants to see, the benefit of aging and health span. Many of the clinical data are not so convincing. So it may suggest there are additional molecular mechanisms inside. This drive us to understand why damaged mitochondria are accumulated and whether they could be a risk factor of Alzheimer's disease if it cannot be a sufficient or independent uh, driver. So, and then we used mouse models, APPPS1, triple TG, APPS1 bearing A beta, triple TG bearing both A beta and tau pathologies. We use these postmortem brain tissues from Harvard Brain Bank. We also worked with Professor Zam Kader in Oxford using their different familiar Alzheimer's disease iPSC as well as ApoE3 and ApoE4. So all the data collected together showing reduced mitophagy in our model systems, which leads to accumulation of damaged mitochondria. When mitochondria are damaged, it reduces the function, activity, as well as resilience of the neurons. Additionally, there are supporting cells in the brain, like microglia and astrocytes. They consider as a garbage, they have the garbage cleaning capacity to reduce A beta. And we find microglia's capacity to reduce A beta was low, And in addition, it releases pro-inflammatory cytokines. But with our strategy to increase mitophagy, we can reduce all of them if we cannot fully stop them. And finally, we saw improved memory in our animal models and improved neuronal function in iPSC-derived cortical neurons.
Okay, so mitophagy is the breakdown
Damage the mitochondria.
of damaged mitochondria. And so it's just like autophagy, like the mitochondria get broken down, and then I guess the pieces are still used in the cell and rebuilt into new things. So how do you
So I think give
encourage? Yeah,
one comment
yeah.
if because people will be reading this. So the most stringent definition of mitophagy is not just to recognize and degrade the damaged mitochondria. It can also recognize and eliminate superfluous mitochondria, as perfectly exemplified by uh, the uh, extinction of sperm-based mitochondria once the sperm meets the eggs. Because for all our mitochondria are from our mother. The, the mitochondria in the father, within, I think, a few minutes after it meets with the eggs, the mitophagy status comes out to reduce this. Our colleague, I think in University of Colorado Boulder, he had a lot of work in C. elegans and have evidenced this. Another example is there is no mitochondria in red blood cells. So a protein called NIX, N-I-X, it is important for the cell to recognize and degrade the mitochondria in red blood cells. In addition to, except those like rare situations, the majority condition is mitophagy to recognize and degrade damaged mitochondria and eliminated by lysosome and finally released to the cytoplasm for cell to reuse.
Okay. And
Mm.
how do you encourage mitophagy?
There are both pharmacological approach and a genetic approach. So why people need to do exercise? Because exercise can increase muscle volume, especially weight based. And when we get older, sarcopenia is a key syndrome of aging. But when we exercise, we increase not only the volume of the muscle cells, but yeah, I think that's because we have more mitochondria as well as more healthy mitochondria inside. So physical activities can increase mitochondrial size and function. We also can go with pharmacological approaches like small compounds, NAD plus precursor. So we were among the first one to show NAD plus precursor is able to increase mitophagy. There are many pathways involved because NAD is so fundamental. One pathway we were involved in was NAD plus is used by 31 to deacylate FOXO3. And FOXO3 is a master transcriptional regulator, can increase the transcription expression of many genes like LC3, ATG12, and NIX. Those encoding proteins involved in general autophagy and mitophagy. And then there was another study as in 28 or 2010 in PNS by our professor Torin Finkel. So Torin had a paper in PNS showing NAD plus is important for the deacylation of many proteins like ATG1, ATG8, which is LC3. Those deacylation of those proteins are essential before they can be formulated as a double membrane factor, the pre-structure of autophagy to recognize and engulf the cargoes.
The lysosome is used for autophagy and mitophagy. Is there something, I mean, so does encouraging autophagy automatically encourage mitophagy, or are they kind of separate?
Yes, so I want, yeah, it's so I want to give a definition. Um, Autophagy, if you consider as an umbrella, it is composed of three types. The number one and most common studied and spoken are macroautophagy. And the second type is microautophagy. Normally, they do not have the double membrane factor to recognize, just can be through different way to be recognized by the lysosome. And the third one is pioneered by Professor Anna Maria Curto from I think Albert Einstein University in New York, called chaperone-mediated autophagy. Most often, when you read the literature, we are talking about macroautophagy, started from the generation of double membrane factor and with to recognize the cargoes and close it, then fuse with the lysosome to have the cargo to be degraded. And the original concept was many of the so when now we're talking about autophagy, I'm specifically talking about macroautophagy until I specify whether it's chaperone-mediated autophagy or microautophagy. So speaking of this macroautophagy or autophagy, based on the um, like the the type of the cargo, we give a lot of subcategories. So if the cargo are mitochondria, we call mitochondrial autophagy. If the cargo is ER, which is ERphagy. If the cargo is a portion of the nuclear, like the lamins, that's nucleophagy. If the cargo are the aggregates, they call agriphagy. So mitophagy is a subtype of macroautophagy.
Right. And so
Mm.
you're right. And the damaged dysfunctional mitochondria are recognized, put
By
into the
mitophagy, yeah.
by mitophagy. Okay.
There are many interesting molecular mechanisms. So there are two I can share with you. So uh, for Parkinson disease, the Parkin, there were Japanese colleagues among the others showing a protein called pink1 and its matched ones Parkin. Those two proteins are important for one specific type of mitophagy. When one mitochondrion are in high quality, you have the mitochondrial membrane potential. So they can recruit cytoplasmic available pink1 to the intermembrane space of the mitochondria. Because they have good mitochondrial membrane potential, and through different ways, it can be imported to half to the matrix, half to the inner membrane potential, inner membrane space, and they are degraded by the enzyme. So the pink1 parking pathway cannot continue. However, when one mitochondrion is damaged, for example, um, there's a very low or even no mitochondrial membrane potential. Once pink1 is recruited, it can be stabilized, then can phosphorylate the downstream proteins like parking, then to initiate the pink1 parking dependent mitophagy pathway. So it's so interesting.
So one of the things you're studying is Alzheimer's, and you have a funded trial that will look at urolithin A as a mitophagy inducer.
Yeah,
Could you talk a little bit about urolithin A and how you can use it in Alzheimer's?
yeah. So uracil A itself is not available in pomegranate. its precursor, which is available in pomegranates and other fruits. Once our body consumes the precursor and our microbiota degrades it to and produce uranacin A, uranacin B, uranacin C, I would say a very classical paper published again by Professor Johan Oben's group in EPFL, showing uranus A was able to dramatically extend lifespan and also healthspan in the roundworm C. elegans. And that was published in Nature Medicine 2016 or 17. So that got our attention because we were working on a big project on whether pharmacological upregulation of mitophagy can reduce memory loss in Alzheimer's disease animal models. Definitely, we use NAD plus precursor, including NMN, but we also want to use other mitophagy inducer to consolidate our hypothesis. So we picked up uranacin A, another one called actinoline. That was actinolin's mitotic capacity, inducing capacity was reported by um, the NIH professor, which I mentioned um, just now,
Think, Finkel?
Torin Finkel,
Finkel.
Torin Finkel's lab, yeah. So using those three different mitotic inducers, including uranacin A, we showed that it was able to induce mitophagy in neurons, increase memory in both uh, the roundworm C. elegans based Alzheimer's disease models, as well as in Alzheimer's disease mouse models. So that paper was published in 2019, and we over the past 6 years, we got effusive and positive responses from the society, as evidenced by its very highly cited, and it was highly cited papers in Nature Neuroscience, over I think 1,500 citations over the past 6 years. So we were continuing trying to get the grant to do clinical trial. And we're very happy in collaboration with our colleagues from Charles University in Prague, the Czech Republic. We got a very decent funding supported by the Alzheimer Association, a US-based private foundation, which supported 4 clinical trials this year. We was only one outside of the USA to get funding. And in this clinical trial, we aim to do a phase 2 double-blinded placebo controlled clinical trial. We aim to recruit 60 Alzheimer's disease patients based on the MMCE as well as A beta and the tau parameters. And we will have 40 blinded to be treated by uranacin A for 1 year, with the other 20 to be treated with placebo. And after 1 year, both of the two groups will continue to treat with uranacin A for half a year more. The reason to design in this way is because Alzheimer's disease is so terrible disorder, and we wanted to increase the incentive of the participants to join our clinical trial.
Yes, I was going to ask about because the thing is, Alzheimer's progresses really quite slowly,
Hmm.
so it's difficult to get sufficient. Will 1 year be long enough to get sufficient
Hmm.
data?
Uh, it develops very slow, but if one pick up the right population, that can really make something. If you use the US FDA recently approved anti-A beta antibody as drugs for Alzheimer's disease, even it was only able to reduce 25 to 30% of the 35% of the speed on the memory loss. But if you look some of the phase 2, I think it's not so long, so it is reasonable. And
Okay.
for our case, our primary endpoint is p-tau 217, uh, a biomarker pioneered by Oskar Hansen in Lund University in Sweden, showing this molecule not only to be detected in the CSF but also in blood. That makes it less invasive. And the blood p-tau 217 seems correlates very well with Alzheimer's disease. So that is the biomarker for us to use. Because our hypothesis is those biological markers may be more sensitive to 1 year treatment than the behavior like the MMCE, those memory-based performance. So our other secondary parameters, including microglia inflammation, as well as NAD plus, because we want to test our hypothesis, is the mitophagy induction, NFL, neurofilament, this fragment. So we have a couple of parameters.
So what dose of urolithin A will you be using?
It is 1 gram per day, a single time per day. This is based on published studies from Amazontas, a company in Lausanne, the Switzerland. They are our financial collaborator, which provides pharmacological level urinalysis A for our clinical trial. Before our collaboration, they have already finished a couple of clinical trials, performed pharmacokinetics, toxicity, as phase 2A clinical trial on muscle atrophy. So it shows 1 gram per day uh, has a very good benefit.
So, so we're kind of talking about trials, and this is a phase 2 trial.
Mm,
So thinking about translation into the clinic,
mm,
how would you, how do you approach translation for something like NAD precursors or even urolithin A?
mm.
Because I think like the phase 2 is, you know, it can be funded, but how could you, will you be able to fund a phase 3 or,
Mm.
and kind of what is the end point that you would look for? You're looking for a like an FDA approved drug, or you're looking for something that people can take,
Mm.
like a supplement.
The latter is not way is the material providers their wishes. For example, Amazontas, which provides uracil A, the NR provide Chromedex, and I think they have no strong intention to making them as a drug because of the time and the challenges. They may prefer as a diet supplement due to different reasons. But for us scientists, we do not care that too much. We cannot control that side too much. What we can do is to using our experiments to show whether they work or not, whether they safe or not. Um, and I think if the phase 2 clinical trial on using nice A, if the results are very positive, for example, after 1 year, we can see dramatic reduction of p-tau 217 and improvement of the memory. We should be able to convincing the big farmers as well as private or public foundations to support us, but that is very expensive. Um, and as you mentioned NAD. So because NAD has been sold as a supplement in the USA for many years, in Europe it's still a very gray field. Um, and there are a lot of noises on NR and MN, which one is better, which one is more stable. and some clinical trials showing the NR-based clinical trial worked on their endpoints. Some showed no benefits. So there were a lot of noises in the field, and every time when we're giving talks, not only just the general population, but also the professionals like the doctors, they also need such information. Which is the current clinical evidence on NAD in clinical trial? Where, what disease it shows it may potentially work, what disease it may not work, what are the potential side effects, what are the doses. So with all those in mind, we were very fortunate to be invited by Nature Aging to commit this commissioned expert opinion with both NAD basic researchers and clinicians to publish this clinical-based NAD expert opinion, which was published in Nature Aging on Tuesday, Tuesday morning at 11 a.m. London time. So it is online now. And we were very fortunate to get the ideas also as co-authors like David Sinclair, Eric Vedin, and many clinicians from Norway and other countries to have a consensus. So our conclusions are, I think it's very convincing. NAD plus precursor works very well for those aggressive premature aging disorders because that was based on three different institutes. For other diseases, for example, Morton Schreiber Colossian, my former colleague, we worked with Dr. Bob together, and he is now an associate professor in Copenhagen University. A few months ago, he published a paper in Nature Aging showing that NAD plus precursor NR was able to partially reduce inflammation-related profiles in COPD. It may not reduce all the inflammatory side effects-based endpoints, but some of them. And another colleague from Bergen, a second largest city in Norway, he already finished an early stage clinical trial on NR for Parkinson disease, which was published in Cell Metabolism. And in this one, he also, I think the key thing is used this non-invasive NMR. He was able to show with NR treatment, the NAD plus levels in the human brain was increased. And in addition, he saw reduction on some of the inflammatory profiles in Parkinson disease patients. Now he's doing a big cohort for Parkinson disease. He's now also leading Alzheimer's disease. And I am also involved in NAD plus precursor to treat for Huntington, to treat for um, the disease Stephen Hawking got.
ALS?
ALS, yeah.
Yeah.
Actually, in Norway, we have a high potential, we have a high instance of ALS. So we are, that has been started for, yeah, some time. So we are excited to see how the results will look like.
Yes. I mean, just one last thought on this. The, the, the thing is, if you do like the Urolithin A study and
Mm,
you show good results, good efficacy,
mm,
without going all the way to get like full approval, you Amazentis or whoever sells Urolithin
mm,
A can't say on the packet this helps with Alzheimer's, even though
no,
your study shows it. And so it kind of falls in that gap in the middle, which is
mm,
unfortunate. Um,
mm.
it'd be good if there was a way around that.
But I think in the modern times, the general population, they are more knowledgeable than we may expect it. If they are interested in their own condition or disease or anti-aging, they follow up, the publications, the social media, and through those channels, if they are lucky enough, they can also get some accurate information.
Yes, I mean,
Mm.
which is why I
That's
do
what you
this.
are doing also, I think. Yeah.
That's right. That's why I'm doing it.
Yeah.
The thing is, I think it reaches a very narrow population,
Mm.
um, and yeah, other people are, more cautious and may not take it. But it is what it is. So as we kind of finish,
Mm.
what are the things that you're working on now? I mean, we talked about the Urolithin A, but what are the emerging technologies, and what's your lab working on next?
Um, we have a basic molecular mechanism, half translational. So we are still interested in, what are the drivers of biological aging? And in the brain, especially for Alzheimer's disease, we are interested in what are the molecular mechanisms that can cause neural degeneration, the death of the neurons. We are working on apoptosis, necroptosis, so different approaches, and try to ask why those like pathways are malfunctioned when we're getting older, especially in Alzheimer's disease. And speaking of translational, we are interested in combination. So actually, we have a paper in preparation, working on NAD and exercise, see whether they have any additive or synergistic benefits to the Alzheimer's disease models. We also have another paper in preparation working on NAD and time restricted calorie restriction. So NAD plus CR, see whether any joint benefits. Those are some of the works we are doing now. We have more than 30 projects in the lab.
There is a lot to do.
Mm,
So one thing I think about, like, Alzheimer's is multifactorial, and I think it's called uh, diabetes type 3. So
mm,
the brain has difficulty getting in enough enough glucose, enough food. So even if you give the brain NAD and it's helping its mitochondria, could it still have problems if you're not giving it enough food? I mean, would like a ketogenic diet or something
mm.
like that help?
Yeah, my former joint postdoc mentor, Professor Mark Madison, he is considered as a pioneer on fasting and exercise as potential intervention approach to slow down the instance of dementia. So, and the ketone diet, I think those are all more or less there with some preclinical things and some of the clinical things. And because of the difficulty, including budget as well as the time, so we do not have so much high quality and large size clinical things to really convincing us, see, this is the way. So I think that is what is missing now. We have a lot of studies in mice to show those approaches work. But from translation to mice to humans are very different, not totally different. So we all want to be accurate, do not want to over interpret it or over promise our potential listeners. Yeah.
Yes, absolutely. Okay, Dr. Fang. So if people want to follow your work, where should they go? Yeah, we can. And can you send me a link to that new paper you wrote? That sounds very interesting.
Yeah, I think if people are interested in our work, you can go to the evandrofanglab.com. I hope Richard will put it at the bottom of your video so they can
Absolutely.
follow up the projects, publications, and different things.
Okay, Dr. Fang. Thank you so much for joining us today.
Thank you so much, Richard, and let's keep in touch.
Yes, absolutely.
Yeah.